Hi everyone, my name is Rizwanullah, and today I'm going to represent this research article is naturally derived by active compound and nanotechnology enhanced formulations, which is related to medicinal chemistry. This research article is published in ACS Journal on 2024. This is the outline of this presentation. First one is introduction to natural product, challenges with natural product, and role of nanotechnology. Second one is material and method, and then its extraction and isolation techniques, bioactive compounds overview. The third one, result and discussions, means nanoformulations, advantage, mechanism of action, and the fourth one, challenges and future perspective. And the fifth one is conclusion. So, first of all, we will talk a little bit about the introduction of natural product. As we know that natural product have historically been important to humankind as a source of medicinal drugs, but also pose challenges for drug discovery and development. We know that less than 1% of the world biodiversity has been truly investigated for potential biological activity. This suggests that many more valuable natural products with beneficial property remain undiscovered. Recently, clinical investigations have been focused on natural products, especially antibacterial and anti-cancer drugs. Although natural items are typically overlooked, compounds laboratories are being created in part resemble the chemical features of natural product more accurately. Natural products have long been recognized as key participants in the drug development process, particularly in the area of infectious in cancer therapies, but also in the field of cardiovasculars, rheumatoid, diabetes, etc. But with this, we have a lot of challenges like poor bioavailability due to limited explorations of diverse ecosystem due to geographic, political, or logistical barriers restrict the discovery of novel natural product. Along with this, we have also a challenge of its low stability. As we know that many natural products are chemically unstable. They are chemically unstable to temperature and pH, and also they are evaporated in nature. They degrade quickly under light, heat, or varying pH conditions, making storage and formulation difficult. The third challenge is variable efficacy. The therapeutic effect of natural product can vary due to difference in cultivations, harvesting time, or preparation method, which affects their clinical reliability. The third challenge with natural product that we have to face its standardizations. Ensuring consistent quality and concentration of active compounds across batches is challenging, especially for herbal and traditional medicines. So, to overcome these challenges, the scientists introduce a new terms technology which is known as nanotechnology. These nanotechnology play a significant role in overcoming many of the challenges associated with the natural product. So, first, the role of nanotechnology regarding these challenges. So, when we introduce these nanotechnology, or means that when we extract or isolate compound from natural product like plant, animal, or microorganism, and then we make its nanoparticle due to which they increase their biological activity and also overcome their low stability. So it enhance the drug delivery and improve its bioavailability, enable the targeted therapy, and reduce its side effect. Means that some alkaloids they are toxic. So when we make its nanotechnology with the help of which we can decrease its toxicity. So before the formation of nanoformulations, we do extraction and isolations. So what is extractions? Extraction basically is the process of separating a specific compound or group of compound from a mixture such as plant extract, tissue, sample, or waste material. Extraction basically yield a crude extract that still contain various impurities and other component. After extractions, the extract are further subjected or isolations. The main aim of isolation is to obtain a more purified fractions containing the desired compound and subjected for further analysis, characterizations, and applications. So for this extraction, we use basically different method like traditional method. In traditional method, we use maceration, chocolates, and percolation. They are all the traditional methods. And we can also use the modern techniques like ultrasonic, supercritical fluid extractions. And in terms of analyticals techniques, we use PLC, HPLC, and GC and MS. So before extraction, first we prepare the sample. Like suppose we take an extract from the natural product like plant, and then we dried the sample and then convert it into powder and then mix with the sample. After that, they are subjected to extractions, and these are the method, the traditional method of extraction, and these ones is the modern techniques of extractions. And after that, these extractions are further subjected for isolations, and we use different techniques for isolation like TLC, column chromatography, HPLC, GCMS, and after that. So after the FTIR Here is the different method for the synthesis of nanoformulations. So the first one is the dendrimers, which are a highly branched tree-like macromolecule used in nanoformulations, particularly used for drug delivery, gene therapy, and imaging. And here is it, it is the method of its synthesis like divergent growth methods, and it is the benefit of these nanoformulations. The second one is polymeric nanoparticle. Polymeric nanoparticles are solid biodegradable colloidal particles made from natural or synthetic polymer. They are also widely used in drug delivery imaging and also in vaccine formations and dispersions of perform polymer is the method through which these polymeric nanoparticles can be synthesized, and it is the benefit of these polymeric nanoparticles. And the third one is liposomes. Liposomes are a spherical vesicle compound of one or more phospholipid bilayers surrounding an aqueous core. They are widely used in drug delivery, vaccine, formulation, and cosmetic due to their biocompatibility. And the method which are used for its synthesis is dispersions, ultrasonications, phase inversion, temperature. And it is the benefit of these nanoformulations. The fourth one in micelles. Micelles, the fourth one is nanoemulsions. Nanoemulsions are kinetically stable colloidal dispersion consisting of oil, water, surfactant, and sometimes a co-surfactant with droplet size typically ranging from 20 to 20 nanometer. They are also used in drug delivery, cosmetic, food, and agrochemicals. And here is its methodology, and it is the benefit of this nanoemulsion. The last one in micelles. Micelles are also a nano-sized spherical aggregates formed by the self-assembly of amphiphilic molecule in an aqueous environment. It is used in cancer therapy, gene delivery, and diagnostic. And dialysis and oil in water, they both are its synthetic method, and it is the benefit of these nanoformulations. So here is the bioactive compound overview. It means that when we extract or isolate a bioactive compound from natural product, then they have a biological activity like polyphenol have an antioxidant activity, alkaloids have anti-cancer and analgesic, terpenoids have antibacterial and also have an antioxidant activity, and like this one cannabinoids have anti-inflammatory activity. So mechanism of action means that how this molecule act in a body of living organisms. First one is a interact with protein and a nucleic acid. Bioactive compounds can be interact directly with proteins and nucleic acid, influencing a wide range of biological functions such as gene expression, enzyme activity, and signal transductions. Cell survival. These interactions are crucial for their therapeutic effect and mechanism of action. Second one is antioxidant and anti-inflammatory pathway. Some of the molecule have an antioxidant activity and they have the scavenging property. And also this induction of apoptosis and cancer cells and means then the bioactive compound which have the ability to cancer cells. So this one is the clinical trial and applications. So here is the clinical trial and its applications. It means that when we extract and isolate the biological compounds from the medicinal plant or microorganisms or animals, then we check its different biological activity and also its clinical application. So some of them are used as a for breast cancers, some are used as a diabetes, and also for different infectious and cognitive diseases. There is a conditions and there is the drug derived from this plant. This so this the main one is these carcolines. So when we make the nano formulations of the medicinal plant, then we further check its activity that either it's mm, improved or not. So it's the plant and is the part of the plant means seed and it's the silver nanoparticle and it is the study model means that it is used for breast cancers. It's also another plant and the researchers use its leaves and make its nanoparticle and use again different microorganisms. So all of these are the plant and this one is the plant part and this one is the nano formulations and he use again this nano again these microorganisms and here is the advantage of nano formulations means it enhances the solubility and stability of the bioactive compound. It's increase the targeted delivery, reduce toxicity. As I told you before, that there are many organic compounds which are naturally toxic, and when we make its nanoparticle, then its toxicity reduces. And here is the sustained and controlled release. Example: liposomes, nanogels, polymer, nanoparticle. This one is nanogels, liposomes, polymeric, nanoparticles, as we discussed already before. So here is the case study of curcumin. Curcumin. Curcumin is basically a bioactive compound. Turmeric is a well-studied natural product with a potent antioxidant and anti-inflammatory properties. Natural curcumin show poor solubility, low absorption, and they are unstable, and they have limited therapeutic efficiency and less than 1% bioavailability. But after nanoformulations, curcumin show high stability, show good therapeutic effect. So here is the challenges in future perspective. Scale-up difficulties, regulatory hurdles, potential toxicity concerns, and need for more clinical trials. And here is the conclusions. So natural product hold massive potential in therapeutic nanotechnology great improve their applications. In future focus means clinical translations, safe nanoformulations. And thank you.